Or, yeah.
Okay.
Got it. Perfect. So it'll record and then we'll have it on our side. Perfect. Okay.
So you think that I can start sharing my screen now or not yet?
Let's wait until the introduction and then I
Okay.
think we can
No
start
problem.
sharing. We'll just give
Okay.
people a second to file in.
Okay.
Okay, I think we can get started here. Welcome everyone to the Atlanta Botanical Gardens Science Cafe. It's a monthly featured science talk, and we bring different specialists from around the country, around the world, to talk about their research. And so today we have a very special guest in which I'll let Lauren introduce, but both Lauren and I are part of the conservation and research team at the Atlanta Botanical Gardens, and we're hosting Tatiana for today. So a big welcome.
All right. Thanks everyone for joining. So I'm so excited to introduce Dr. Tatiana Arias to you today. Tatiana is a classically trained botanist who also uses modern methods. She has 15 years of experience in plant science. Tatiana has a PhD in biological sciences from the University of Missouri and spent 2 years in a postdoctoral fellowship at the University of Hong Kong. She's also worked a lot in several institutions and universities in Colombia, and Tatiana's research integrates multiple disciplines, including systematics, genomics, collections-based research, and natural history, all with the aim of understanding the amazing diversity in the orchid family. And so with that, I'm super happy to have you, Tatiana, here today. So I'll give you the floor.
Okay, thank you very much. So let me share my screen first. And I will go from there. All right. Are you seeing the right screen? Okay, sounds good. Well, first of all, I wanted to say that I feel really excited to be here. I am starting several collaborations with the Atlanta Botanical Garden, and particularly with Lauren. And it's an amazing opportunity to be able to share a little bit of what I do, and particularly what I do in Colombia. So thank you so much for inviting me today. So yeah, we are going to talk today about orchids of Colombia. But before we do that, I just wanted to talk to you about and explain a little bit about some of the reasons why Colombia is so diverse or has so much biodiversity. And then my talk will be divided in talking about orchid diversity very briefly, and then what I do, and particularly in science. I have been working in this position for 2 years, so I'm gonna show you a lot of preliminary data. And then last, what I want to tell you about is about big project that we have in Colombia that is called Orchids for Peace, and that has been going on since 2018. So I wanted to start my talk giving you a very simple explanation of why Colombia might be so diverse. And as you can see in this slide, let me just use the pointer here. Okay, as you can see in this slide, Colombia is located in the northernmost corner of South America, right? And is this area is located in this area of the planet where more sunlight is like there are more sunlight in the planet, right? And it's also, Colombia is also strategically located between Central, or it's the connection between Central America and South America. And more or less 3 million years ago, there was a great exchange of biota between Central and South America. So those are some of the reasons, but when you also look at Colombia, you will also see an amazing geographical diversity. So in Colombia, the Andes branch, when they enter Colombia, they branch in 2 different cordilleras or mountain ranges. And we have part of the Amazonia, part of the Orinoco, the Atlantic coast and the Pacific coast. So all these reasons might have allowed for Colombia to have so many different species, okay? And people often ask me what Colombia looks like. And honestly, like a simple answer to that question doesn't make justice to reality. In my opinion, there are many Colombians inside Colombia. So we have people from many colors. We speak, or within Colombia, we speak more than 65 indigenous languages. And we have, as you can see, a ton of different orchid species, bird species. We have dense jungles and paramos. We have, again, as I say, the Caribbean Sea and the Pacific Ocean, and savannas, mountains, rainforests. I can keep telling you all the things that we have within Colombia. And all this makes that our diversity really, really great in terms of like culture, but also biodiversity, right? So this slide I brought here just to show you a little bit of the representation of like number of species that we have in different organisms, and in many of them, we, in such a small area in comparison to other countries like Brazil, we have a huge amount of species, and we are supposed to be one of the countries with more bird species, mammal species, and of course, orchid species. And getting into the orchids now, and to talk a little bit about orchids of Colombia, or orchid diversity in Colombia, today I want to focus in this particular group that is a group of genera within the Orchideaceae. And this group is called a subtribe, and the name of that subtribe within the Orchideaceae, within the family Orchideaceae, is called is PLEUROTHALLIDINAE. So this is perhaps the group that we have that has more orchid species in Colombia. There are perhaps 2 or 3 genera here that are not included within this group, such Epidendrum and Maxillaria, and some other groups that are also super diverse. But in general, these, as you can see by the number of species, this subtribe has a bunch or a very big portion of a percentage of the number of species of orchids that there are in Colombia. So that's the group that I am going to be talking about today. And these are some of the genera that are very species rich in Colombia. You know, as you can see here, for each of these genera, I am showing you the number of species. And I'm also showing you not only how rich they are in terms of species, but also how high the endemism within these groups are. So you can see here that a big portion of the species that we have in Colombia, we can only find in Colombia, and within Colombia, many of these really endemic species or these endemic species are also really pretty restricted to different locations, to small locations, I mean, to small areas, which makes the orchid or this group in particularly very susceptible to changes in the landscape. And the second thing that I wanted to mention about Colombia, and this is true for like the Andes in general, is that most of the diversity that you see in orchids or orchid species is gonna be located in the cloud forest. So this is like high elevation mountains, and usually they are most of the time covered by these horizontal precipitation, as I would like to call it. This basically is when like the vegetation is receiving like significant amount of precipitation, like mist, that is intercepted by epiphytes or like orchids, bromelets, or all plants. Epiphytes are all these plants that grow in a specific host or tree, but they are not parasites. You know, they live there, they complete their whole life cycle there, but they are not parasite in the trees where they live. Those are the epiphytes, and many of the orchids that we have in Colombia are epiphytes. So let's talk a little bit about what I do and what I am researching in Colombia. So I'm particularly interested in 2 genera. The first genera that I'm going to talk to you about today is the first genus that I'm going to talk to you about today is Lepanthes. Lepanthes are these really tiny little miniature orchids that not many people can see in the forest because they don't have like a trained eye to see them. But once you start to identify them, you won't miss them, right? So Lepanthes means scale in Greek, epis scale, and anthes flower. Okay, and this is the flower of, or the inflorescence of Lepanthes. This is one of the flowers. You know, in Colombia, as I told you, there is more than 1,000 species, and I could tell you that probably every other week somebody is describing a new species of Lepanthes in Colombia. These plants grow, as you can see here, from like 50 meters of elevation all the way up to 3,600 meters. Even though you like the majority of species that you find above the 1,500 meters of elevation. And as you can see, they are widely distributed in the neotropics from Mexico to Bolivia, and we have some very few species in Guyanas and Mexico. So, and this is a pattern that we are gonna find in these orchids that I am gonna be talking about today, is that they are basically characterized because they have these like all these endemic species that have like really specific preference for habitat. And again, this makes these plants really susceptible when there is like deforestation and habitat lost. Also, when there is like random disturbances in the landscape, and when, of course, people come and poach and grab these orchids to take them for illegally sell them or for growing them in their houses or whatever reason it is, they are highly susceptible to this. The populations seem to be really small, although we don't know if that's because many of them live in the canopy, and we don't have access as easily to the canopy as we have to the lower, like the understory and the other portions of the forest. And then about the floral structure of Lepanthes, I can just tell you that these are called the sepals, the ones that are here that I am showing you. Lepanthes has two bilobe petals, and this is the lip, which plays a very big role in pollination biology and attracting the pollinators. Okay. So the first thing that I want to talk to you is this study that my student from Universidad de Caldas in Manizales, Colombia, has been working on for the past year. And what we really wanted, or what we wanted to do with him, was to sequence the chloroplast. You know, the chloroplast in plants is maternally, in most plants, is maternally inherited. And this chloroplast, as you can see here, has a ton of different genes that we can use to reconstruct the phylogenetic history of this genus. So, and at that time, what we really wanted to do, we have a very, very tiny budget to do this. But what we really wanted to do to start working on this was to take the taxonomic classification. This is like a taxonomic classification that Carl Luer, the botanist that actually work in Colombia with many Colombian botanists and was a researcher here at Mary Selby Botanical Garden. This botanist did this classification, and we have one of the most spectacular Lepanthes collections in the world, I would say. So he basically, a taxonomic classification is just taking the species and using phenotypical or morphological similarities. In this case, it could be in the flowers, as you can see here. And then what the taxonomists do is, based on all these morphology, they make groups, smaller groups, and then bigger groups. So basically, our initial hypothesis was that we wanted to see if these, let's say, morphological similarities, floral similarities, will actually reflect the evolutionary history of Lepanthes. So what we did is that we picked one species for the different like subgroups, taxonomic subgroups that he made, and then we sequence, and I will go back to this, we sequence all these genes for each of these species. And then with that, we were able to reconstruct what we call a backbone of the phylogeny of this genus, right? And what I'm showing you here, so this is the taxonomic classification. This is our final tree that we got with the different like categories, or each species representing the different taxonomical categories. And what you see here is the the morphological or the floral variation of each of these plants, representing each of the groups of species. So what we found, so again, remember, Lepanthes has more than 1,000 species, and this is not at all a very good percentage of the species included in this phylogeny, but this could give us at least some like a starting point to see if the variation in floral morphology had anything to do with the evolutionary history. And we did recover different clades that make sense. As you see, Lepanthes rivets and Lepanthes felis are grouped together, and they kind of have a similar floral morphology. But some other groups, they have been grouped together in this taxonomic classification, but they are not grouped together here. Like if you see things like Lepanthes mucronata, I don't find it here, and Lepanthes monoptera, they don't belong, or they are not sister species, or they are not forming what as we call clades. So with this information, what we could do too is to, once we have all these genes within the chloroplast, what we are doing now is figuring out which of these regions is going to be the most informative to reconstruct big phylogenies that use 300 species, 400 species, and things like this. And there is only one study that has tried to do this with few chloroplasts and nuclear markers, but they didn't get any resolution. So for us, it was really important to see which of these markers will be more informative, so we could use the correct markers to see if we actually will get a good phylogenetic story or not. Okay, so that was like the first step on this research, and this is where some of the data that I have come from. But I'm also working with a lot of taxonomists in Colombia that are very good and know really well the natural history of these species and the morphology and the classification, and they recognize pretty much all of these species, and they can say when a species is new or when it's not. So one of these taxonomists that we are working with is Sebastián Moreno. Sebastián Moreno just finished his master's degree at Universidad del Valle. And what he did was that he put together this database with all the distribution of Lepanthes in Colombia, and using geographic information system program. What he did was to map things like endemism, as you can see here, or richness, like species richness, species endemism, and to see where in Colombia there was like more species diversity and where all these endemic species were located. This also was a very important resource for us to plan the next study that I am going to talk to you, because these areas pretty much told us where we should go next and what we should sample next. So I want you to remember here that Antioquia is a big region, this region right here. You know, and I also want you to remember El Valle and Nariño, all these parts over here. And I also wanted to tell you that there are some places still in Colombia where we could not go, and still we cannot go, like Norte de Santander. It's really dangerous there. And also Santander. So the idea was that once we have phylogenies, we can combine the information that Sebastian has gathered to understand if mountain uplifting has something to do with this richness and endemism, or if things like pollination and pollination syndromes have to do anything with these patterns or geographical patterns that we see. So this is the part of the story where Lauren and the Atlanta Botanical Garden come in. So in, oh gosh, I forgot, like 2021? I'm sorry, Lauren, I don't remember. This paper came out in Applications in Plant Sciences, and basically what Lauren and other friends or colleagues did was to develop this kit to sequence many different loci or many different genes across the genome of several epidendroid orchids, which Lepanthes belong to that group of orchids, right? And then they develop all this really great method to actually capture more than like 900 genes to be able to reconstruct evolutionary histories that could make it, or that could be much more robust. So I reached out to Lauren and I talked to her and say, like, we have this project, we want to work with Lepanthes. Would you like to join us? And she did, and now we have a great partnership, not only with Lauren, but also with Sebastian and my colleague Alejandro Zuluaga from Universidad del Valle, and many other people that I am not mentioning here. And the idea is that the first step that we did to start this big project or this big endeavor was, again, look at Sebastian maps, I'm sorry, and figure out where we should go in Colombia to collect the samples. And then here we went. So I have this really beautiful experience for a month in 2021 too, in which we went to all these different regions. I took my car and I went with students. Different students were meeting me in different places while I was going down south in Colombia. And in total, we collected more than 145 species of Lepanthes. We found in this expedition 6 new species that we are describing at the moment, and we keep finding more and more. We visit around 10 Colombian departments, and we're in really pristine forests. And yeah, and did field work, but we also went to do some, we visit some living collections, and I'm going to talk to you about this because this is a very fundamental part of my research too. But before that, and because I need to drill this into people's mind, into botanists' mind, into conservationists' minds, I wanna talk to you about the importance of collecting vouchers of orchids, right? Of course, not everybody has to be collecting vouchers of orchids, and not all of us have to go to the same areas. But just by being here at this institution that has so many orchid collections, I can also see the gaps that we have in collections that we need to fill in the next few years, because species are also disappearing. So there is like a, you know, a need to do this, but to do this in a very conscious way, right? So once I go to the field, I have figured out that I want to collect a very nice herbarium specimen. Perhaps this one is a bit too much, too many plants here. I think that with one will have been enough, but this wasn't like, I don't know, in the 80s where people didn't care about this. And then I make sure that I also do a collection in what we call like a spirit collection. That is a collection that has alcohol, glycerin, and water. Why? Not all plants need this. You know, many of the plants that some botanists study, they can do with things like working on like with the characters and with the things, the features that you see in the herbarium specimen. In an orchid, you cannot do that. You need the three-dimensional, like the three-dimensional arrangement of the flower to see and to be able to identify it. You know, and so that's why you really need to have a spirit collection of the flower. And we do have, like they gave me the number today, we have around like 2,000 spirit collections only for Lepanthes. We have the biggest spirit collection, orchid spirit collection after Kew in the whole world here in Selby, at Selby. Besides collecting these, I'm also collecting photographs of the same specimen, right? Of the same specimen in many different angles, and then tissue for DNA studies. So this is my voucher, and this is the voucher that if you are gonna collect orchids, please do too. Okay, we could also talk about how to label this, but I guess that I'm not gonna take that much time talking about that anymore. And as I say, we also visit like local nurseries because in some cases they are the best source of germplasm. You know, so there is this guy, he's also an Lepanthes specialist, and he has this amazing private collection in his farm of all these circuits that are well labeled, that have some of them have good geographical information, but this was very pivot, like very important for this study, because we were able to get many collections from him. We also were to Andrea Nissen nursery in Valle del Cauca, and then Ramiro Medina in Putumayo. I work with these collectors on a regular basis. Some people prefer to fight with them, and some people prefer to tell them or to accuse them that they are poaching orchids from the field and all this, but I don't think that that's the right approach for conservation. So I do work with them, and I use the collection as source for scientific and conservation, scientific research and conservation. So the other thing that we want to do with this data so where is this at the moment? At the moment, we are trying still to get these collections out of Colombia. So taking orchids or orchid collections abroad is a really painful procedure. You know, you have to have many different permits, and we are still, that was at the end of 2021, we are still trying to get samples out of Colombia. And we'll do it, we'll make it. It's progressing, but it's progressing really slowly because of bureaucracy. And once we have this here, then all those samples are gonna go to Lauren, and she's gonna do her magic so we can reconstruct the phylogenetic relationships among all these species that we have collected and all these samples. And once we have that, then the fun part, well, the field work part is really fun, but the fun part of like answering questions such, why are they so diverse? Is the environment making or is it shaping all the diversity that we see out there? How is the history related to the diversification and also the changes in the environment during like millions of years? You know, and there is a ton of different characters and morphological characters that have been used to explain this species radiation or species explosion in many orchids groups. So this is what we do next. But to do that, we need to have robust and big databases too. And this is where my awesome volunteers and students come to play a very big role. This is Luke Rominecki. He's a high school student that has been working with me for a year. And he's been doing amazing work because he's extracting pretty much, he's like converting all the taxonomical descriptions into like data, into tables, you know, and files that then we are going to be able to use with these phylogenies. And this is just to show you what he is doing. So far, he has scored more than 34 characters for more than 177 species. And this is a very preliminary analysis that I did with this data, like a principal components analysis on all the perianth parts, or like the floral parts, to see how they relate in this like statistical way. And each of these dots is a species. So you can see that most of the species are grouped together, but you can kind of have some outliers outside that space. Nothing to interpret here yet. I just wanted to show you what we are doing. Okay. So a second study that we have in Colombia that is also progressing really slowly is this study. So before I go there, I wanted to say that one of the most fascinating facts about orchids is the relationships that they have with other organisms. And to me, what is more fascinating still is how they have relied on these organisms so much that pretty much they have, like orchids have given away huge responsibilities to pollinators and to fungi, to basically like these responsibilities like reproduction and germination. So in that sense, what I wanted to tell you is that orchid seeds and orchids need a fungi, or need mycorrhizae to germinate, be able to grow, to germinate, and to be able to establish into the environment before starting to do photosynthesis. So, or before it started to feed itself, you know. So what mycorrhizae does for orchid seeds is to provide the nutrients in the first stages of development, right before they start producing their own nutrients, right? So this is probably my favorite group of orchids ever. These are called Masdevallias. They are also rare, super diverse, endemic from Colombia, etc. And in particularly, I like this subgroup within the Masdevallias, I mean, that is called MELEAGRIS. It's also a group that LUER, like a taxonomical group that in which LUER put all these species. Most of them, in fact, are from Antioquia, that region that I show you, where I am actually also from, the northern part of Colombia. And they look like aliens, right? And they are, most of them are also rare and endangered. So the first question that we have, and that we are still waiting to have samples here too, is how these plants are related, whether they do share a common ancestor or not. And we have also gotten like root samples from each of these plants. We have isolated mycorrhizae. We are amplifying, or we are identifying molecularly and morphologically all the mycorrhizae that each of these orchids had. Of course, this has a very fancy experimental design and all this that I'm not going to mention here. And then the idea is that at some point we are going to be able to identify those mycorrhizae, be able to isolate these fungi, and be able to mix the seeds, the orchid seeds, with the mycorrhizae in things like these tea bags, and be able to reintroduce these plants into nature to do like some augmenting programs. So this project is also in progress. The last project that I was going to talk to you about, and I don't know if I'm going to have time to talk about anything else after this, but is this Dracula phylogenomics project. This project is also ongoing, and it's also we are also working with the set of baits that Lauren has. And in fact, Lauren and her team are working right now on the DNA extractions and sequencing of all these samples. But let's start from the beginning first. So it's been really painful to bring Lepanthes samples here from the field. So it occurred to me that we could use, again, private collections from people living here in the US to study some of these orchids. Of course, all these plants, they have acquired legally through nurseries that have brought plants to the US legally, right? And in particularly, I became really friends with Mary Jo Saxes, which is this lady right here. She's an orchid nerd, and she really loved these Draculas. You know, Dracula means small dragon, and you can see that they are these really odd balls orchids. Again, rare, endangered, all of these things that you already know. But what Mary Jo did is that she was able to recreate all the conditions that these really specific, really specific conditions that its orchid needs in her house, in a closet room here in Florida. You know, that's like mind-blowing to me. And she did it all by herself. She figured out all the systems that she needed, the light, the humidity, everything that she needed. And now she has a very amazing Dracula collection at her place that we have available to do research. The second person that I want to mention here, and these are the conditions that Draculas need, so you know how pretty impressive having a collection like this here is. This is Aaron Fent. Aaron is a retired surgeon, and he's the one recording or making all these amazing photographies for me. We are, again, collecting, doing all the herbarium records, making amazing pictures that one day we hope we can publish in a book, things like this. So as you can see, I have a lot of great volunteers working here with me. So I want to tell you a little bit about Dracula. Dracula is not as diverse as Lepanthes, but it's pretty. It has a bunch of different species that they go from Mexico to Peru. Of course, the majority, so from these 140 species, we have about 90 species that we are sequencing at the moment. And of course, we have a very huge diversity or a very huge percentage of this diversity in Colombia. And Draculas, they have pretty interesting pollination systems. They have this lip or this floral part that looks like a mushroom, and unfortunately I don't have really good pictures of that here, but you see this is the lip. It looks like a mushroom. It attracts fungus flies that probably come and pollinate the flowers, but we don't know much about the pollinations of these flowers, but it seems like, yeah, they are imitating mushrooms to bring in flies that pollinate them. One other thing about Draculas is that they are really ill-defined as species. I mean, most of species in orchids are very ill-defined, but these in particular, there is a lot of hybridization going on, hybridization not only in the greenhouses, but also in nature. So this phylogeny is going to help us to understand a lot of different aspects of this genus. Okay. And as always, I only have 5 minutes to talk to you about my outreach and community experiences. So there is no need to say that orchids are the pandas of plants, as I like to call them. They are super charismatic. They attract a bunch of people, community scientists, foundations. And that's one of the things that I like the most about orchids, actually, to be able to interact through them with people, you know. And that's how this project became what it is today, Orquídeas para la Paz. And it happened when I visit Caquetá. Somebody invited me to give a talk at a university there. And actually I met Lauren and Emily over there, I think, for the first time. And I met this woman, Mari Polania. You are gonna see her over and over. And she was an orchid fan, a plant fan. And she also told me her personal story. And then I started thinking about this project that has become a flagship project for the region, for me, for the gardens, you know, so in which we combine research, conservation, orchids, and communities, right? And then the first place where we started to do this is in El Manantial. This is Florencia, Caquetá, a small city in the Amazon Piedmont with the Andes. And this reserve is a community reserve, or a community reserve that is located in the outskirts of Florencia. And this is basically this project. You know, what we have is like, I have students, like graduate students and undergraduate students from Universidad de la Amazonía. And we wanted to do basic science, you know, we wanted to do exploration and inventory in a region that was like really, really touched by violence. And that was like actually starting to be more open to everybody as the things were improving in Colombia, and the peace agreement, after the peace agreement was signed. And then we did, and then with this exploration, we wanted to prioritize orchids with horticultural potential, you know. And in the other hand, we wanted to give all these collections that we were having, living collections, to bring them in these community nurseries for reproduction, and also for this community to use it as a showcase for bringing tourists into trails, but also into the actual nurseries that we were building. And with that, we wanted to provide this community also with trails and with training and with field guides and things like this. You know, so yeah, 5 or 4 years later, we have done a bunch of expeditions, but we haven't covered, I wouldn't say more than 30% of the territory. So we have a lot more work to do. But like 2 weeks ago, this first paper came out, a big collaboration between many students and researchers, like taxonomies and other researchers from Colombia. Like the first checklist of orchids of Caquetá. So I feel really proud of this, and I feel really proud of working with all these people together to make a very important contribution to the region, you know. So besides that, we have this amazing living collection at El Manantial, and these living collections have many orchids from the region that we have been reproducing vegetatively and in vitro for 4 years. The collection keeps growing. There is a lot of students and volunteers that come to help us to transplant and to vegetatively reproduce this material and things like that. We have done all sorts of travels around Caquetá, met with a lot of communities trying to communicate what we do and to teach them how to take care of orchids. Many of my students are now super involved with this, super involved with orchids in the department, and I pretty much go there and I don't have to do much because they do all the work, and it's awesome to see them being the trainers now of other people. Yes, I say we did a ton of workshops, and now we are in the process with another volunteer of mine that knows how to do all these things and manage photos and all this. We are gonna do, and this I took from a friend. We are doing these like field guides of orchids that are like rapid field guides. And we are gonna do it by like all the genera from Caquetá first, but then we are gonna go deeper and do the like genera like Epidendrum and Maxillaria. We are gonna do a smaller field guides like this for the public, you know, and these are gonna be in Spanish. Okay, so I think that I'm gonna leave this right here. The only thing that I wanted to say is that we have done research within the nurseries. Yeah, like taking a lot of data about flowering times, about doing auto-pollinations, and these are being taken to the in vitro cultivation. We are also starting to do DNA extractions for all these orchids, and the idea is that we will sequence that. Very quickly, I want to show you that we did this interpretative trail in this community reserve where people go to different stations, and all of them have been growing their own like orchids and giving pieces away to friends and taking them to other farms and things like this. This is orchids and people, you know. We also built this like maloca with medicinal plants for some of the people, for this indigenous, for this Witoto indigena there. And we have created a bunch of like field guides, books about medicinal plants, about traditions of like histories and tradition of the Witoto, and some like manual for like orchid cultivation and in vitro orchid cultivation. In the region, we have also done a lot of like participatory sciences using iNaturalist. And now our project is expanding. So I'm working with Jaime Góngora from University of Sydney in making nurseries for ex-combatants around many regions, or about several regions in Colombia. And you can see that within the university and the botanical garden in Florencia, they are also making their own orchidiario now that they call PLEUROTHALLIDINAE. Isn't that cute? Anyways, sorry to go that fast through all that, but I don't have more time. And basically, what I want to conclude my talk just telling you that our studies in Colombia aim to understand the origin and evolution of species-rich genera in the Orchidaceae, while we are promoting also conservation inside and also outside the country, because we are going to start bringing, and we have started bringing a lot of living collections for ex situ conservation here at Selby. This is the effort of many people. I'm just like communicating all the hard and good work that a lot of people are doing in a very awesome team. Some people have also given us some money, I have to say. And that's it. With that, I would take any questions that you might have.
So you can ask questions in the Q&A box, and I'll moderate them and read them out to Tatiana. Thank you so much for your talk. It's really fascinating to see all of the amazing work that you've done over the span of your career, even just with communities in Colombia. It's quite incredible to see how involved they are in your research. That was great. Yeah, Emily Coffey said, thank you very much, Tatiana. Amazing talk, great work. Thanks, Emily.
Hi, Emily.
Yeah. Lauren, actually, well, we have a huge storm going through Atlanta right now, so some people's power are going in and out.
Yeah, I think that we are going to have a big one too.
Yeah. Okay, we have a question here. I was wondering, with orchids being so fragile and climate specific, have they been affected at all by climate change?
Yeah, that's a great question. So we have many orchids already included in like the IUCN red endangered orchid list. But I don't think that it is enough. We still have a lot of data to collect and analyze, and we need as many people as possible that are involved and that want to get involved in helping us understand what the actual state of the orchid population, of the orchid species, like at a wider scale, not only Colombia, but like the neotropics in general. And I talk about orchids in the neotropics because that's what I do. There is also a ton of species diversity in Asia. But yeah, in order to know how affected these orchid species are, we need to be able to get together with many researchers from all over the neotropics, but many of them are pretty endangered, but more evidence is needed to make like conclusive how do you say this, like very solid conclusions about those things.
Okay, thanks. Here's another question from Brian. Does the limited genetic diversity of selectively bred orchids impact your research when sourcing materials from other collectors?
The impact my research. I would ask this person in which sense. I would say no. Like if I do a lot of like genetic analysis and like molecular and genomic analysis, we find that when we have less variation, it's easier to do many things. So when you work with crops, for example, and just to give you an example, so when you work with crops, you want to have like these things that are called double haploids, or that are like homozygotes all over the genome. So you have less variation, so you have less noise, and maybe you are simplifying your stories, but life is so complex that that's the easiest way to take the next to understand something and then take the next step. So I don't know if that answered your question, but I hope it does.
Okay, great. And then we just had one other comment here. Just Bob was saying how impressive the diversity in Colombia, considering how small it is, even just compared to the size of Texas, and how much diversity there is there. So that was his comment, but no more questions coming in, which is probably good timing. I just wanted to say thank you again, Tatiana, for your talk. Next month we have Dr. Hector Rocha coming in from Mexico. And so he is a scientist down there in the Cuatro Ciénegas area, and he'll be talking about the secrets of the origins of life. And so he's an ancient microbialist, not him himself, but the microbes he works with. So he'll be coming into the gardens next month, August 17th, and we'll be in Mershon Hall. And with that, I'd just like to say thank you again, Tatiana, for your time, for your expertise, and for your collaboration here at the gardens. We're still very excited to continue working with you, especially with the Dracula project. And I'm sure Lauren would say the same if she had power in her house. So
absolutely. Okay.
Yeah. Thank you very
Thank you.
much. Okay. Bye-bye.
Bye.